Today, I will discuss AIONER, an all-in-one scheme for biomedical named entity recognition using deep learning by co-first authors Ling Luo and Zhishuan Wei, with Po-Ting Lai, myself, Robert Leaman, Qinyu Chen, and our PI, Jiong Lu. Biomedical named entity recognition, or NER, is the task of identifying biomedical concepts such as genes, diseases, chemicals, and many others in text. Biomedical NER serves an important foundation for many tasks in biomedical natural language processing, enabling entity linking or normalization and relation extraction. These tasks together support many of the downstream applications of biomedical natural language processing, from information retrieval through discovery of new knowledge. The importance of NER has led to increased attention in recent years, and modern transformer models, particularly those based on BERT, have become the de facto standard due to their consistently high performance. But despite being pre-trained on unannotated data, these models still require annotated data for fine-tuning to the end task. Unfortunately, this data is scarce. Even if we intentionally overestimate the availability of human-labeled corpora for biomedical named entity recognition by pooling all of it and counting documents with at least one annotation, only roughly 0.1% of all the articles in PubMed would be considered annotated. The scarcity of this data implies that the majority of the variations in the literature will not have been seen in fine-tuning, limiting generalizability. However, most corpora are annotated with only a few entity types, fragmenting the available data and further reducing generalization. It also makes creating systems which can identify multiple entity types difficult. The most straightforward solution, creating a separate system for each entity type, dramatically increases resource usage and can also cause errors when the results are combined. One possible solution is multitask learning, or MTL, which shares the hidden layers of the deep learning model across related tasks while maintaining an output layer specific to each task. MTL can improve the model's generalizability by leveraging domain-specific information found in the training signals of related tasks. However, the performance improvement of MTL isn't universal across all datasets, and MTL often requires a complex model architecture. Our approach considers an alternative to modifying the model. Namely, we focus on modifying the data. To explain our approach, we must first briefly mention the tagging schemes used in named entity recognition. The simplest tagging scheme uses the label O for tokens outside of an entity and an I or for inside labels for each entity type. The most common tagging scheme adds a B or begin label for the first token of each entity mention. More complex tagging schemes like IOBEW introduce E or end labels and W labels indicating single word mentions. The AIO, or all-in-one tagging scheme we propose, adapts the IOB tagging scheme to account for corpora which are only annotated with a subset of the entity types to be identified. The top sentence is from a corpus which only has gene annotations. The bottom sentence is from a corpus with annotations of all 6 of the entity types of interest. Let's look into the details of the AIO scheme. First, the labels for entity mentions are the same as in the IOB tagging scheme. Second, the O or outside labels are now differentiated by the type of corpus, either a single type or all. Finally, a token is appended to the start and end of each sentence in the training data to signal the type of annotations which are expected. Our AIO scheme is enabled by the BioRed dataset, a recently developed corpus of 600 manually annotated abstracts from PubMed articles. This dataset is annotated with all entity types of interest and serves as a bridge to integrate the other datasets, which are annotated with only a subset of the types. Our AIONER system proceeds in 4 stages. The first 2 steps collect the 10 corpora used for training and unify them into a single collection using the all-in-one tagging scheme. Then we train a deep learning model on the data. This work primarily uses PubMed-BERT, a biomedical BERT model pre-trained on abstracts and full texts. That model can then be applied to documents which were previously unseen. We compare AIONER against the baseline of training on only the bio-read training data and also multitask learning. Evaluation on the bio-read test set demonstrates performance improvements against the baseline on all entity types. AIONER also outperforms MTL for all entity types except genes, with MTL's performance even dropping against the baseline for species and cell lines, suggesting that the performance improvement of MTL is indeed somewhat task-dependent. AIONER also consistently outperforms both the baseline and MTL as we decrease the number of bio-read abstracts used for training. Evaluation across the test sets of all training corpora revealed similar results. AIONER proves beneficial on average, with performance dropping compared to the baseline on only one dataset. AIONER also outperforms MTL on average, and MTL performance drops compared to the baseline for 3 datasets. We further assessed AIONER on 3 datasets with new entity types outside of our AIONER training data. Here we use AIONER in 2 ways: merged, which adds the new data to the AIONER training data and retrains, and pre-trained, which uses the trained AIONER model and then further fine-tunes it on the new data training set. Both methods produce consistent performance improvements compared to the baseline and to MTL, while MTL's performance improvements are task-dependent. We also performed an analysis of different model variants, considering both the efficiency for 100 abstracts and F-score for the bio-read test corpus. We see no significant differences in efficiency between PubMed-BERT and BioBERT, but we do see significant time savings with Bioformer with the softmax layer, while only affecting the F-score about 1%. The AIONER approach helps for 3 primary reasons. First, AIONER can better categorize the entity type based on the context. For example, the mention HCV genotype 1 infected is incorrectly recognized by the baseline as a species, but AIONER can correctly detect it as a disease. Second, AIONER also presents higher accuracy in detecting the entity boundaries. For example, in the case of Vogt-Konágy-Harada VKH syndrome, the baseline detects the wrong boundaries and identifies it as 2 entities, while AIONER correctly identifies it. Third, some entity spans that are not shown in the training set may be missed by the baseline, but AIO can handle those unseen entities better. Although AIONER exhibits promising performance, there are still some errors. 36% of the remaining errors are incorrect boundaries, mostly dealing with whether the leading or trailing tokens should be included in the span. For example, whether necrotizing should be included in the disease mention necrotizing fasciitis. Second, entity type ambiguity accounts for almost 27% of errors. This error comes in 2 types. Mentions like growth hormone refer to a protein, but this mention might be annotated as a gene or a drug, i.e. a chemical, depending on context. Or the mention may potentially refer to multiple concepts. For example, BMD may refer to either the disease Becker muscular dystrophy or to the gene which causes it. There are 2 relevant limitations. First, the AIONER approach requires a dataset like bio-read, which is annotated with the entity types of interest. However, we have shown that AIONER can be successfully applied to additional entity types and datasets. Second, like other straightforward tagging schemes, the AIONER approach cannot directly handle overlapping or disjoint mentions. In conclusion, our AIO tagging scheme allows the model to be trained on more diverse datasets. This allows the model to learn more synonyms, which improves generalization. AIONER provides performance improvements more consistently than multitask learning. It is also straightforward to implement and should work with any deep learning transformer model. I would like to thank my co-authors, particularly co-first authors Ling Luo and Chishuan Wei, also our PI Jiang Lu, and the collaborators who made the BioRed corpus possible. The source code, trained models, and data for AIONER are freely available online at GitHub, and the BioRed corpus is also freely available via FTP. Any questions? Please feel free to get in touch.